So we have obviously the ZUMA-1 study, which again will be further reports about that at this meeting and in publication soon. What is the indication, obviously, that led to approval again of Axi-Cel or Axi-Cel last in October?
Right. So we're tremendously excited to have an approved CAR T-cell for lymphoma, and Axi-Cel was the first one, or Axi-Cel, and the results for the study was basically treating 101 patients with a CAR, and they had to have had relapsed refractory diffuse large B-cell lymphoma or diffuse large B-cell lymphoma transformed from follicular lymphoma, predominantly are the two main groups. And in the clinical trial, actually, they had to have even higher risk disease and either relapsing within 12 months of an auto transplant in other populations. The actual label is 2 prior therapies and relapse refractory disease, and that's the way that the label now indicates. And the bottom line is about 50+% of those patients had complete remissions, and they appear to be fairly durable. The data we have in the PI label was through, I think, 8.9 months or 8.7 months of follow-up, and we're anticipating data at this meeting looking at about a year to year, 15 months or so of follow-up. And everyone's quite excited because it appears that these remissions are quite durable. But that's still a question about the field. Is the early work from James Korkendorfer and Steve's earlier work suggests that patients who have a complete remission can have durable remissions, and I think some patients are out of 5+ years in those studies, and so this is tremendously exciting. And I think you have to remember, these people are not even eligible for an auto transplant, or they'd already failed. So this is a population, you can't say, oh, we'll rescue them with a transplant, because they couldn't either get it or they already have failed it. So this is a population that has a significant unmet need and just tremendously exciting results. The treatment was pretty well tolerated, but we talked about the toxicities. Cytokine release syndrome, neurotoxicity were significant. Almost half of the people required some type of intervention with either an IL-6 antibody receptor antibody blocker or with steroids. And so I think we have to temper our enthusiasm by that, but the results, I think, speak for themselves. I mean, there is nothing else that would give this kind of a remission in this population. So tremendously exciting. It's being opened in about 12 centers, I think, 11 or 12 centers initially, and that will greatly expand probably to 100 centers over the next years. And I think so. This should be thought of as a treatment option earlier on in the case of these patients' disease. I don't think you have to wait, till 5th, 6th, 7th, phase 1 study. This should be moving up into, and it's an improved setting. So, second relapse disease should be treated. Obviously, as we talked about, the question is, can we threaten auto transplant, right? Will CAR T cells do away with auto transplants? And it's possible. The studies that are kind of going forward will be taking people at first relapse, randomizing them to CAR T cells or transplant in some iteration of that. And so it's very possible that if these results if they pan out and hold out in an earlier setting, that may be better than what we can do, even in the chemosensitive population where, what are our results? It's about 50% best case scenario with an auto transplant. Right, so.
No, and I have to add my own enthusiasm. I have to say that we were one of the higher accruing sites to the ZUMA-1 study. I did see patients who were absolutely refractory, would certainly not have been eligible for transplant. We were even considering hospice, who had pretty remarkable responses. Dangerous to talk an anecdote, but I think that the aggregate data do, justify that enthusiasm. And again, I think for those of us who are in the transplant community and are interested in cellular immunotherapy, I think that the trials have to be done. We have to obviously confirm the persistence of responses and look at that. I think that, but the data are looking very encouraging. These curves on the successive iterations do appear to be flattening out, as they did in prior studies from the NCI. So I share that enthusiasm, and I think that this is really a good time for all of us who are interested in I think, these diseases and lymphoma and immunotherapy in particular. So I want to talk a little bit about the nuts and bolts of this, again, for those who are not familiar with this. We do an apheresis, we prepare the cells, and then we bring the patient in before during the manufacturing process, right, or when the cells arrive back at our site, and give them really some chemotherapy, right? So we give what we call lymphodepleting chemotherapy, and that typically consists of fludarabine and cyclophosphamide.
Yeah.
So you want to talk about
I
the
think
reason for
it's
that?
a key point. A lot of people don't quite understand what the heck are you giving Cytoxan and fludarabine for, because these people are already chemo refractory, and often we would not anticipate that amount of Cytoxan and fludarabine is going to cause any kind of major response. And in fact, that's true. The reason that lymphodepleting chemotherapy is given is to actually reduce the number of endogenous T cells. And when that happens, the body makes signals and cytokines to actually stimulate T cell production. And that's when we want to introduce the CAR T cells to then allow them to engraft better and expand. And Dr. Korkendorfer also at this meeting has studies, as do we, showing that if you give this lymphodepleting chemotherapy, you actually will increase those levels of serum cytokines such as IL-15, and then that correlates with the expansion of the CAR T cells. We have done experiments sequentially using just Cytoxan or then adding fludarabine, and by adding fludarabine, we got much greater T cell depletion, much better cytokine production, and much better T cell expansion. So we think it's very critical to do this, and it's a theme that is seen generally across the CAR T cell trials, although I think Steve used slightly different regimens in his trials.
Yeah, and I think that from a biological standpoint, that it was actually shown in HIV-infected patients and patients post-allotransplant that when you have that lymphopenia, you have high levels of both IL-7 and IL-15. That drives actually NK cells, which are not relevant here, but also T cells, and I think that that's exactly right. So the fludarabine and cyclophosphamide reduce the endogenous number of lymphocytes. They drive up those cytokines that help to drive now, the expansion, which is also antigen-driven. They reach their target, they expand, and that these cytokines help to drive that.
Exactly.
So I think that this is exciting, and so I think we talked about the fact that in the trials versus the label indication just says really 2 prior therapies. So would you agree that that's a little bit broader than what was done in the ZUMA-1 study? And Steve, as a lymphoma person, do you think that that's where you would refer somebody?
I think that the label is broader than the types of patients that were treated in the study, and I think that we have to remember there's incredibly exciting results. We're saving lives that we couldn't have saved just a few years ago, and I have a tremendous amount of enthusiasm for this. However, this is gene therapy, and we have 5 years of follow-up. We don't have 15 years of follow-up, and so I would just sort of caution people, if we have therapies that are successful for patients that have been around for a long time, we should continue to use those, okay? And if somebody, at this point in time, if somebody was a transplant candidate, I think that that's the standard of care. So I wouldn't say because somebody had a relapse after R-CHOP and got some Rituxan and didn't respond, they had 2 prior therapies now, and I would move on to this. I don't think we're there yet.
So let me add, I think, to that. I think that one of the things we like to say in the transplant community, because we look at the number of patients with high-risk AML, that we would argue that no academic hematologist would disagree about whether a leukemia doc or a transplant doc, that many of those patients actually just never get to the center. Right. And we think that we know that there's a dramatic underutilization. So the one thing I would say is the community doctor doesn't necessarily have to make a decision about whether it's appropriate to treat that patient. In fact, that's a very subtle and complex discussion. But what they should do is at least refer for consideration.
Yeah,
don't you?
Yeah, absolutely. I agree with that.
And they can look at clinical trial opportunity and look at the commercially available products.